Hello, this is part 2 on Factorial ANOVA. It's going to break down a little bit more information beyond the conceptual into how the computational components of Factorial ANOVA are actually constructed. We're still, though, at a conceptual level, and we'll be saving calculation for problem solving in a class session. So first, to compare the Factorial ANOVA with the one-way ANOVA. When we had the one-way ANOVA, we had, besides error, so setting apart error, which we then called the mean square in terms of a variance component or sums of squares within, we have our between groups comparisons. And in one-way ANOVA, you only have between groups comparisons. And in Factorial ANOVA, you would call that sum of squares A, where your only independent variable was variable A. In a two-way Factorial ANOVA, that means you have two independent variables, your sum of squares between is more complex because it includes the between groups of factor A differences, the between groups of factor B differences, and then the between all possible combinations of groups, which is the interaction effect. So all three of those things, A, B, and AB, all are sources where groups may be different in a systematic way due to some aspect of the intervention rather than just to random sampling error. So they're all part of the between differences. And you can look at that in terms of sums of squares in that your total, going down to this equation at the bottom, your total sums of squares, your total summed of squares deviation from the means are composed of the deviations between groups on factor A, sum of square deviations between groups on factor A, sum of squares deviation between groups on factor B, that's SSB, SSAB, which is the variability of each, the variability of each cell mean around the grand mean. And note that in SSAB, where it's explained above, the unique variability of each cell mean AB around the grand mean, you actually remove the A and B effects. So it's basically like, how much do the combinations of the two factors matter significantly, irrespective of the factors of the contribution of the general trends. So it's unique variance associated with that, with those contingencies or interdependencies. And when we do actually in effect sizes for Factorial ANOVA, you can see this effect size in terms of additional effect related to the interaction, and it also will come up more in regression. And then the last compositional component of the total sums of squares, which relates to total variability, is the sum of squares within, also called sum of squares error, which is just the variability that is, as it were, naturally occurring within cells, which is expected to be random. People were randomly assigned to a cell, so their variability isn't expected to have any systematic source. Then a graphic way of doing this is you break your sums of squares total into between groups comparison, between sources comparison. Let's talk about source of variance, between sources of variability and within variations not attributable to a systematic source that would be your independent variable. And then with a two-way ANOVA, you're going to break down your systematic variances, or which derive from your sums of squares between, into the sources of the two independent variables. In two-way factorial ANOVA, you have two independent variables, the source A, the source B, and the interaction. Whereas the error term is just error no matter what. So look at these equations, they look horrible, but they're not as horrible as they might look. One thing is that they add this level to your term for the grand mean, which is the double dot. And that's because you have you have two kinds of group means, and one of them is summed over the other two. Okay, you have, I'm not going to say this right, so I'm just going to show it. And got to make sure I'm sharing right, because it doesn't look to me like I'm sharing right. I just need to pull over a screen. So in your first equation, this looks a little different because of the two dots. It is exactly the same as sums of squares total from before. It's the sum over all the cases over all. J is one group as it was before. J is one independent variable. K is the other independent variable. So it's all the people within all of factor A and all of factor B. It's their individual deviations. So if this person is the first person within the delayed feedback condition and the peer source, then that's their I, first person, J, delayed condition, K, peer source, or whatever I just said. And it is deviated from the grand mean of all means. Every single one of these individual people scores is deviated from the grand mean of all means. Each of the deviation scores are squared. They're all added up for the entire sample. Okay, so now we have sum of squares for the first factor. This is assuming, all of these equations are assuming a fully balanced design. As I said before, even conceptually, much less computationally, if you don't have a fully balanced design, this is all unnecessarily complex. I mean, it's necessarily complex for the analyses, but it's unnecessary for teaching purposes. So we're assuming that the number of people in a cell is constant. And so what this J dot is, so I kind of said J is the timing condition. So this is the mean for each J group averaged over all of K. So deviated from the grand mean. So you're going to have one mean for immediate, deviated from the grand mean, and one mean for delayed, deviated from the grand mean. And these pay no attention whatsoever to which source of feedback they're in. They're a main effect for time. And so this is, going down one row, going down a column, paying no attention to column values, just the mean for a row. And the mean for the first row is delayed. The mean for the second row is immediate. Either one of those is averaged over all conditions of K, the source of feedback. Each of those means is deviated from the grand mean. You take all those squared deviations, summed, you multiply them by the number of K conditions, which is self, peer, and teacher. So you'd multiply in our example by 3. And you also multiply by the number of people in a cell. It's very congruently, I think that's really the word that I'd use, but still. Here you have the mean of each K condition. So one K condition is self, one K condition is peer, one K condition is teacher. Those means are calculated regardless of J. So you calculate the mean for self, including both immediate or delayed. This is a column score. And you deviate it from the grand mean. The column score for self, the column score for peer, the column score for teacher. So those are each 3 means that are being calculated without attention to their row affiliation. Each of those 3 means is deviated from the grand mean. Each of those deviations is squared. They are summed. You multiply by J, which is the number of rows. That's 2, and the number of people in each cell. Okay, now this truly looks horrible unless you notice that this term was already calculated and this term was already calculated. So what you're doing is you are calculating a cell deviations from the grand mean, cell means, not individual scores, cell means from the grand mean. And you're taking out those parts of them that are solely attributable to the 2 independent variables. So the mean JK is the mean for a cell. Say you have 10 people in each condition. Then it's the mean verbal expression score for those 10 people who belong to delayed self. Another one would be those 10 people who belong to delayed peer. It's each cell mean. So it's a mean for both J and K. It's deviated from the grand mean. Each of those scores, and there will be 6 scores because factor A has 2 conditions, immediate and delayed, and factor B has 3 conditions. So it's a 2 by 3, which is going to give you a rectangle with 6 cells. So there are going to be 6 of these squared deviations. You add them all up and you multiply by the number of people in each cell. And then you subtract these 2 terms that you've already calculated. Last, you have your within or your error term, which is the sum of the squared deviations of each individual score from its cell mean. So it's the within variance. It's the variability, not variance, sorry, sum of squares. It's the variability or spread within a cell. And because you have random assignment, you assume that that is just random or error, that you don't have anything systematically causing that one. You might have something systematically causing delayed versus immediate. You might have something systematically causing the scores in the source of feedback or even the interaction. But within a cell, you have no reason to think that those people they will have variability, but that it will be other than random, no. And then your total sums of squares going all the way back to this, that term should be the sum of all these terms that you are able to calculate using the raw data. Okay, so we're almost done here. Your degrees of freedom for a two-way fully crossed analysis of variance is for your, this is pretty self-explanatory. For your A factor, it's the number of conditions of A or groups of A, J minus 1. For your B factor, it's the number of conditions or groups of factor B, which is K minus 1. For your interaction, it's J minus 1 times K minus 1, and your degrees of freedom is and so on. This is, I think, pretty straightforward. These are just formulas. You don't have to memorize these formulas. You know, I always forget where my degrees of freedom, sometimes I'm better than others. I remember, all right, who cares? You can always look these things up. Your mean squares, now everything gets easy. Okay, if you get this far, and we're not going to hand calculate these, I don't think. So I don't think, no promises. But if you get this far, you're good. You calculate the mean squares just as, now it mentions here repeated measures, we haven't covered that, but just as for anything else. A mean squares or a variance source is its sums of squares divided by its degrees of freedom. Just standard. And you have more of them with a two-way ANOVA. If you have a three-way ANOVA, you know, then you're going to have even more, because you're going to have, say you have a 3 independent variables, you're going to have 3 main effects. Then you're going to have an interaction between effect A and effect B, an interaction between B and C, an interaction between A and C, and an interaction between A, B, C. It can get a lot. That's why it's difficult sometimes to interpret a three-way ANOVA. But with a two-way ANOVA, you will have 3 possible sources of statistically significant effects: your first factor, your second factor, and the interaction between them, and you'll have your error term. Each one of these is calculated as a separate test. The F test is that mean squares. For instance, in this case, it's the sums of squares for the interaction divided by its degrees of freedom. It gives you the mean squares divided by the mean squares due to error, which is the sums of squares within divided by its degrees of freedom. It's all once you have all your sums of squares, the further calculations are just follow a standard source of getting mean squares and then using mean squares to calculate your F statistic. There are different critical Fs for each test because the number of degrees of freedom for the effects can vary. Not the number of degrees of freedom within, but the numbers of the degrees of freedom for the effects can vary. The assumptions are, as always, the assumptions are the same as with other assumptions for ANOVA. The scores are independent of one another. The sampling distribution of the means is normal. That does not mean that the data are completely normal, and that you satisfied the homogeneity of variance assumption, which we won't go through any details about how to test, but a test is available in most software packages, including SPSS. And the last thing here is the effect size. The effect size in two-way ANOVA is slightly different than the effect size in one-way ANOVA. It's called partial eta squared. And that's because the denominator term, the numerator term is the same. It's the sum of squares for that effect. But in one-way ANOVA, the denominator is total sums of squares, which lets you interpret the effect size eta squared as a percent. but this is not the total, because that would be leaving out the contribution of the B. That would be, I'm sorry, including the contribution of the B factor, which really you can't count. It would inflate. I'm sorry. It would deflate. It would deflate your eta squared estimate. Yeah, it would deflate your eta squared. Your denominator would be overly large. And so this is really partial eta squared because it's that proportion of the sum of squares that is uniquely attributable to A divided by kind of the portion that was available to be attributable to A when this had been removed. I think this is the sort of thing that is much better expressed in regression, because we talk a lot about accounting for variance, and this has a corresponding term that we'll go into in a fair amount of detail in regression. But partial eta squared, and I'll say it again because I think I'm better at saying it, has to do with the sums of squared attributable to factor A divided by what's essentially the sums of squares total, as with eta squared, subtracting those parts of the total that have been attributed to B for the interaction. So it's, yeah, that's just, I'm going to stop there because I think that was a pretty good way of saying it. And that is entirely where the whole show stops. So I am going to wish you a good evening or afternoon or morning, whatever it is, and see you next time.